to today's Johns Hopkins Malaria Research Institute Friday seminar. It's a pleasure to have Dr. Tony James with us today. He's a professor at University of California, Irvine, Departments of Microbiology and Molecular Genetics and Molecular Biology and Biochemistry. Dr. James received his bachelor's degree and PhD from UC Irvine. He did his postdoctoral training at Harvard and Brandeis University and became an assistant professor at Harvard School of Public Health in 1985. And later on in 1998, he became a professor at UC Irvine. I was a junior graduate student when I met Tony for the first time at the MacArthur-founded Vector Biology Network meeting in Santa Cruz in 1992, and Dr. James has played a very important leadership role in shaping the modern vector biology field, aiming at disease control. One thing that I found very interesting with Dr. James' professional trajectory is that it actually looks like he already knew as an assistant professor that his work would largely focus on developing the type of malaria control strategy he will tell us about today. Early on in his faculty career, he developed a strategic plan or a framework where he defined the essential tools and key components needed for building malaria-resistant mosquitoes, such as promoters, effector genes, etc. He actually isolated and characterized the first mosquito protein encoding genes involved in blood feeding in 1989. And blood feeding inducible promoters. He developed the first transgenesis system for mosquito in 1998. And in 2000, he demonstrated for the first time that it was possible to engineer an anti-pathogen gene to confer mosquito resistance to malaria parasites. More recently, his laboratory and colleagues have developed the first CRISPR-Cas9 gene drive system in Anopheline stevensii, the Asian malaria vector. Dr. James has also played a really important and essential role in preparing the grounds for the first transgenic mosquito releases as part of the population replacement strategy by developing a regulatory structure or framework for working with genetically modified mosquitoes in disease endemic countries that address legal, social, and ethical approvals. He's currently leading the University of California Malaria initiative that is focused on developing anti-parasite gene drive mosquitoes and to identify field sites in Africa at which it will be possible to conduct safe and ethically sound release trials of the population alteration strains. So with that, I'm going to hand over the microphone to Dr. James, who will tell us about population modification of vectors for malaria control. I would also like to ask you to keep your questions to the end. You can use the messaging system to type them, or you can turn on the video at the end of the presentation and take your questions. Thank you.
Well, thank you very much, George. And it's a real pleasure to be able to present to you. George has seen me talk for many, many, many years. And so he already knows that my talks are really just progress reports. And so today is going to be another in a long series of progress reports, but to bring you up to date where we are in our particular program here. I'm going to provide a short introduction. I know that this is the specific malaria seminar series at Johns Hopkins, but there are people from outside of Hopkins who aren't in the malaria field who might need a little refresher on the background here. And I'm going to give you some of the background that we developed in terms of the objectives that we're trying to achieve, and then go into some new data now. Okay, let's see if I can do this without messing it up. I put the acknowledgments page up first because I tend to rush toward the end, but I want to say that this work, of course, is done in great collaborations with members in the laboratory and other institutions. And then what I have across the top here are the institutions who we're active with now, and then some in the past whose data I'm going to show. The University of California, Irvine, Davis, Berkeley, San Diego, Johns Hopkins University, and some previous work that I'm going to show from the Institut Pasteur, and funding that's contributed to that: the Tate Institute for Genetics and Society, UCI Malaria Initiative, Bill and Melinda Gates Foundation, NIH, and the Keck Foundation. So for those of you who are just tuning in casually, I guess, we can ask the question, what is malaria? And most of you know it's an infectious disease, an intracellular protozoan parasite. And many years ago when I gave lectures like this, we talked about 4 species, falciparum, vivax, ovale, and malariae, as being the principal pathogens for human malaria. But we now know that there are a couple of emerging parasites, Plasmodium knowlesi and simium, that are moving out of the animal environment into humans and are causing infections in humans. What you see down here is an image of a human red blood cell smear, and they're supposed to be empty. And you can see that this is the parasites that are in there, and this is the definitive diagnostic for malaria, which is a blood smear. It's a mosquito-borne disease transmitted to humans only by the mosquitoes in the genus Anopheline. And there's a high degree of host specificity. So there are malaria parasites that infect humans, lizards, birds, and mice. And humans won't get rodent parasites, and rodents will not be able to support the development of human parasites. And this becomes important later on as we develop some of the reagents, but there's a high degree of specificity there. And of course, there are no free-living forms. They're either in the mosquito or in their host. Just a brief update from the World Malaria Report 2019. It's still endemic in the poorest countries. There are about 405,000 deaths estimated in 2018, 228 million cases, and most of them are centered in sub-Saharan Africa. And this long, persistent situation with malaria has called for a second wave of people calling for malaria eradication. And so this is a principle under which we're working and trying to contribute. So what are we trying to do about this? Well, we're interested in population modification of vector mosquitoes. And what we want to do is change the ability of the mosquito to transmit pathogens. So we want to break the cycle of the pathogens going from the mosquito to the human host. And we think that this idea of population modification has great value because we feel it could be a sustainable impact on malaria elimination. And that is that one can go into an area and put mosquitoes in there that are resistant to malaria parasites, and then that could be sustained without further releases if they're well designed enough. There's no empty ecological niche, so we're not emptying the niche of mosquitoes. Things that would dine on mosquitoes would still have the ability to do that. They would just be resistant to transmitting malaria. This technology should be stable in low population densities and areas where there's fluctuating mosquito abundance. When it gets very low, our genes should be preserved in the mosquitoes that are still present there. So we anticipate that this should allow for a serial approach in essentially a resource constrained manner. That is, that one can pick an area where this technology might work, go in and clear it of malaria, and then leave with some expectation that that area will remain malaria free. So this would allow you to consolidate gains as you move from place to place. Okay, so our targets are twofold at this point. We're interested in Anopheles gambia and Anopheles stevensii and the transmission of Plasmodium falciparum. Gambia, of course, is a major vector in sub-Saharan Africa, and Anopheles stevensii is in Southern Asia, and more recently now in the Horn of Africa. So how are we proposing to achieve population modification? What we would like to do is engineer mosquitoes with genes that prevent malaria transmission. And the genesis of this actually came out of the genetics of vector competence. And many people have heard me say this before. There are a lot of things that feed on blood, and there are a lot of blood-borne pathogens. And not everything that feeds on blood transmits every blood-borne pathogen. There are very strict host-pathogen interactions, and indeed, they have a genetic basis. And this could be demonstrated in species of mosquitoes, for example, that transmit malaria parasites, when one could go into them and actually select out populations that were either competent to transmit or incompetent, so that within one species, one could get two distinct populations that shared genetic background, but were sufficiently different that some were vectors and some were not. And you could take those two populations and start doing standard genetics and crossing them together. And in many cases, one could show that the ability to transmit belonged to a small group of genes, or in some cases even a single gene. And so that the genetic components of vectorial capacity and vector competence could be illustrated within a single species that way. And very early on, George in his introduction talked about, you know, thinking about this many, many years ago. We thought, wouldn't it be great to get our hands on those genes? Because then what we could do is figure out how to increase their frequency in vector populations, and then we'd have all these mosquitoes that would be resistant to malaria transmission, and you know, we would have achieved this particular goal. But it turned out early on that getting our hands on the naturally occurring resistance genes, at least in the early stages, was going to be very difficult. And so we took an approach now where we decided, well, we will just make those genes. We'll make novel constructs for that. And our ruling hypothesis, or our hypothesis, is that somehow if we can introduce into a mosquito population a gene that would confer resistance to malaria, this should result in less disease and death. So this is a entomological goal in terms of this introduction of these genes, but the readout is actually epidemiological, less disease and death. All right, so in thinking about how one might do this, we thought of three major research areas that needed to be developed. The first one was to show that we could actually make these mosquitoes in the laboratory. So this is molecular genetic manipulation of mosquitoes, focused heavily on the laboratory work. Once one could do that, or have with some success with that, the idea was to figure out how we could introduce these genes into populations. And this would involve both laboratory and field work. And ultimately, of course, we needed to know about the target species in the field. And what we have here are images of three different environments. We have a village in sub-Saharan Africa, a village on the banks of the Amazon River, and an urban setting in India. And they're immediately recognizable as being completely different. And however, they all share the fact that malaria can be transmitted in them. So if one is looking to introduce genes into a population, one has to know about the local ecology. So how are these synthetic genes made? We call it a synthetic approach, but it's more of a collage type approach, and you'll see how that works out in just a second. But we talk about a simple model of a gene where we have a control region which allows when, where, and how much of something to express. And we propose to use mosquito-derived control DNAs or promoters to control as this part of the construct here. And then we have an effector region which kills or disables the parasite. And then of course we need transgenesis. Then if we make this thing, we ought to be able to put it back into the mosquito. So work was done to develop all of these things here. The strategy then is to look at the life cycle of the parasite in the mosquito. Mosquito takes a blood meal, the parasites enter the midgut, they go through developmental stages, exit the midgut, and then make their ways back to the salivary gland before being transmitted. And since we had the luxury of designing our own genes, we decided that, well, it would be a smart thing to do if we're making effector molecules to interfere with the parasite, to put those where the parasites were. So we were interested in control sequences that would allow us to express things in the midgut, and that would allow us to go after these parasite stages that are developing there, the open circulatory system, which is the hemolymph, and then perhaps the salivary glands as they enter the salivary glands. So the first thing to do was to look for genes in the mosquito whose expression profiles would allow us to place something in these various compartments. And we came up, and we and others, whenever I use the word we here, I'm talking collectively about the field, came up with a number of them that are very useful. And this rather garish looking slide has some logic behind it. Here's our midgut, hemolymph, and salivary glands. This is the gene origin. These are the endogenous products of genes and their corresponding genes whose control sequences we're interested in. The color code here actually refers to the expression profiles that we can see here in these images below. These are fluorescent marker genes that are used to validate the function of specific control sequences. In some cases they were green, yellow, blue, and red, and it just happens to have all of them here. Interestingly about these, some of them are constitutive, that is, they're on all the time, and the Tryptophan is one of them. Others are inducible, and they're inducible in females. And most of you know that it's only the female that feeds on blood and is therefore relevant in the direct transmission of malaria parasites. And one of the advantages of this is that these genes are not on in males, so they should not impose a load on the male. And then when they're induced, they're usually induced to a very high level. And so one could use them to boost the amount of your effector molecules in the mosquitoes. And then depending upon which ones you pick and use, your parasite targets can be the gametes, the ookinetes, or the sporozoites. So this is how we build the control package of a particular synthetic gene. The question now, of course, are what are the effector molecules? And there's been a lot of research looking at potential molecules that could have an impact on the parasite. Some are actually parasite ligands, which we anticipate interacting with the mosquito as it makes its journey through, as the parasite makes its journey through the mosquito. And there's a list here, and I'm not going to go through these. I just want to point out there's quite a few of them. And I tried to list as many as I could because I didn't want to have colleagues online now looking up and saying, my gene isn't there. All right, so I tried to be as inclusive as possible. There are tissue recognition receptors. This is the converse here in the sense that these are molecules that are present, the ligands are molecules that are present on the parasite. The tissue recognition receptors are ones that are in the mosquito, and the idea would be to interfere with the mosquito's ability, with the mosquito binding of the particular parasites. And then there was a lot of work, and a lot of great work on looking at immune responses to parasite infection. And indeed, some of those genes that were responsible within species for conferring resistance within a species had to deal with aspects of the immune system. And the idea was that these things could, either as direct effectors or through elevating a general immune response, kill out parasites. So there's a lot of work that was done on these. One could imagine just expressing a toxin, and there's been quite a bit of work on scorpion as something which could be engineered into an insect and kill the parasites. And then there are other modes that also could be looked at. But we were very much interested in the ligands, and the logic behind the ligands were that if we interfere with parasite molecules, we should not be hopefully damaging too much of the mosquito and not be imposing a load on the mosquito. So we're trying to minimize that load. And so we settled for single chain antibodies as parasite resistant molecules. And if you remember, we were talking about host specificity. If you take human parasites and put them into a mouse, the mouse will mount a very strong immune response and basically disable those parasites. And a lot of people were very much interested in this because they could put in specific stages of the malaria parasite into mice and then derive antibodies against those stages. And those were candidates for transmission blocking vaccines. And so we benefited tremendously from the transmission blocking vaccine world. But the complicating thing for us was that these molecules were basically the products of two genes, a light chain, which is here in green, and a heavy chain, which is the orange color. And it's the variable fragment that's responsible for binding to the parasites. So we were able to get a number of these and then clone out the variable portions of the heavy chain and light chain. And the idea, of course, now is to link them to a promoter that would allow the expression of this. And this should look very much like that very simple model of a synthetic gene where you have a control region and an effector portion. And if we're fortunate, we get a single chain fragment variable or a single chain antibody. And depending upon the linker length, it can be either monovalent or divalent or even multivalent. But the idea and the expectation was that this particular molecule would have the same kind of binding properties and perhaps inhibitory products that the parental IgG would have. So these were derived, most of them from mice. So we had to do codon optimization to turn them into things that look like mosquito genes. And in many cases, we could actually link an antimicrobial peptide to these so that we could develop something which is an immunotoxin, so that there would be binding of the single chain to the parasite, but also bring along something that potentially could kill the parasite. So our strategy then is to do compartment-specific expression of dual genes. And our compartments that we focused on were the midgut and the hemolymph. And our target molecules were molecules that were on the gametes, the ookinetes, and the sporozoites. So the idea is we can block them at this stage, or this stage. And we wanted to use 2 at a time because we were concerned about efficacy, that is how efficient they would be, and also about perhaps selecting for resistance. And the idea of using 2 molecules at a time to counteract resistance would be that should you get resistant to one particular molecule, then you could get it at a later stage with the second one. And the probability of giving a double resistance would be very small. So how are these experiments done? One takes mosquitoes and feeds them on a falciparum infected red blood cell culture. We've given them additional meals because remember, some of our promoter sequences are blood meal responsive. That is, they turn on in response to a blood meal. And if you give them additional blood meals, you boost the amount of effector molecule that's in there. And then we dissect mosquitoes after a certain number of days to count oocysts, and then keep another cohort much later on, dissect and look for sporozoites. And an example of one that's worked very well for us over the years is a transgenic line that we made that has dual effectors, one that goes after a chitinase, which the parasite uses to penetrate the mosquito midgut, so it's an anti-ookinete, and an anti-CSP antibody that goes after the sporozoite. And we do a standard sort of challenge assay that you saw in the previous slide. And these are representative data from a number of experiments that were done. And one thing to look at here is that there are 5 experiments. And this is the mean intensity of infection. So this is how many parasites we see in infected animals. And for the controls here, we see anywhere between 2.2, 2.3, small number 2. oocysts, or sorry, oocysts per midgut, and these are the oocysts here. But when we wait and look on the salivary glands, we actually see 0 parasites in the salivary glands. So even though there's a reduction here of about 1 or 2 oocysts, whatever are left do not produce sufficient sporozoites to get to the salivary glands. And so if we have no parasites in the salivary glands, we have no transmission. So this was a very pleasing thing. We worked a long time on this, and it worked out to where we could get this 0 parasites in the salivary gland effect. And we wanted to hit 0 because there's data that show, at least from way back when they could do these kinds of things, that very small numbers of parasites were sufficient to cause infections in humans. And so we went for 0 here. Okay, so I think at this point you can say, okay, you know how to make parasite resistance genes, and you can make transgenic mosquitoes that express them and have this effect. How are they going to be used? Well, one can imagine an inundated release of males carrying these genes. You have your parasite resistance molecules here, sorry, parasite resistant mosquitoes here carrying those genes, and you just cross them to wild types, and with segregation patterns, they'll pick up these genes. And if you keep doing that, eventually all of your mosquitoes should be parasite resistant. You can do another strategy where you couple your resistance factor, which is in green here, with a lethal factor that'll kill mosquitoes that don't have it. You do this type of cross, and every generation, the only ones that survive are wild types and ones that are carrying your effector molecules, and there's a whole class that die. And with time, subsequent releases, they should all turn green. But then there's another procedure, or another approach, which is called a gene drive approach, where you couple your effector molecules to a gene drive mechanism, cross out the wild type, and then all you get out are your resistant mosquitoes. And this is what we were interested in. So what is gene drive? It's a preferential inheritance of a genotype. And this is usually observed in diploid organisms during sexual reproduction. It's been called a circumvention of Mendelian patterns of inheritance. The pattern of the law of random segregation. I have a lot of respect for Mendel, and so I try to be as generous as I can. I don't use the word violate Mendelian genetics, all that kind of stuff. All right, so the drive mechanism is the underlying biological drive feature incorporated in the system, and the drive system is actually the final synthetic product. So there are a lot of mechanisms and transposable elements. Other types of selfish genetic elements have the ability to provide this type of preferential drive. but we were very much captivated, as many people were, by the CRISPR-Cas9 system for developing gene drive. And then we'll talk about that shortly. So the genotypic and phenotypic consequences, if we have a mosquito that is homozygous for a dominant incompetent gene, so it cannot transmit, and it's crossed to a homozygous competent recessive mosquito, you get a heterozygote here. And if you do a test cross, and you would expect to get a 50-50 segregation profile out of this, and half of them would be resistant to parasites and half of them would be not. But if you have a gene drive system, you do the same kind of cross, you get an anticipated heterozygote, but when you do the test cross, you find that they're all the same. Okay, and that's the genetic definition of a gene drive system. Okay, so how do you make this with a Cas9 system? Well, you have on your chromosome a target gene, and that's in blue here, and you build a plasmid that has the Cas9 nuclease encoded in it and driven by a specific promoter. You have a guide RNA which directs the cleavage of this endonuclease to a domain or a specific region in your chromosome. And these guide RNAs are 23 nucleotides in length, and they have some specific features that make them work. And then you have homology arms, regions that flank the guide RNA target site that are homologous to the chromosome location to facilitate recombination. And so these are made synthetically in the insect, and then what this does is it cleaves, and then you get a recombination event. And now you have a primary integration into the chromosome. But because it's carrying the Cas9, the guide RNAs, if you look at that heterozygote, potentially heterozygous animal here, it's carrying the machinery to take this heterozygous animal and cleave that other chromosome and then generate your homozygote. So this is the mechanistic aspect, a schematic representation of the mechanistic aspects of gene drive. And it takes advantage of the Cas9 system, which allows you to get a specific cleavage, and then the cellular biology of DNA repair that facilitates this recombination. So we built one of these. This is just the same thing you saw right here. It just looks a lot more complicated because the target gene has a very specific structure. In this case, it's the kynurenine hydroxylase locus in the mosquitoes. It's involved in the tryptophan biosynthetic pathway, and mutations in mosquitoes give white eyes. And we're talking about work now with Anopheline Stevens eye up here. So this is the gene. So this is the one that's in the chromosome, and this is our plasmid. And our plasmid has the Cas9 nuclease driven by a vasopromoter, which is a developmentally regulated promoter that's turned on generally in the developing eggs of the female mosquito. And the guide RNA driven by the expression of a U6 promoter, which is a promoter that really makes good RNA from that. It carries a DS-red marker gene, and in this particular construct, it carries two of our effector molecules, as well as the homology arms here. And so what we expect to start with is a heterozygote in our primary integration, and through conversion or gene drive, it should go to a homozygote. But occasionally we have a circumstance where it cuts the target gene, in this case the KH gene, and doesn't recombine but causes a mutation. And this is by the abutting of the two ends of the cut chromosome. It's called non-homologous injoining. And this generates a site which is potentially mutant for the target gene and also may be resistant to guide RNA mediated cleavage. And so these are thought to be or could be potential resistance alleles that are resistant to gene drive. So what do we see in a cross? If we do a male out cross, so the male's heterozygous, presumably starting with the gene drive system, we get a circumstance where, so we're just following DS red now. He's outcrossed to a wild type female, and we look at the progeny and we see that there's 3,500 progeny have DS red, all right, and only 48 that don't. And so this is obviously not random segregation. So there's drive going on there. These are the phenotypes. This is the wild type eye. It's hard to see the DS red in an adult mosquito. So we usually score larvae, but this is what the adults look like. The thing that's interesting here is that they have the gene drive locus here, and the progeny you score, but they also have wild type eyes, so this dark colored eyes. If we do the reciprocal cross, where we start with a drive female and I'll cross it to wild type males, we still get, from the first generation, we still get strong drive, 98% here, but the eyes are all white and or mosaic. And so this is the wild type eye. This is the drive animal with that's heterozygous for the wild type. But you can see here now that in these females, they have a white here, and it's obviously much easier to score the DS red there, or they have mosaics, and we can talk about this perhaps in the Q&A, but this is the principal phenotype that they get. Now, if we take females and males from this lineage, okay, so it started out as female crossing, and we take those females and males that come out of this, and we continue to do crosses with them, we find that the transmission and conversion rates go down, meaning that the gene drive slows down. All right, and this happens whether it's a male that's carrying the gene drive and white eyes, or a female that's carrying the gene drive and white eyes. So this is slowing down. So the question is, what's going on? And this is what we think is happening. If the drive loci are coming in from the male side, so this is a male here and this is a female, the female makes the egg, she places her chromosomes in there, and then the male sperm brings in chromosomes that carry the drive locus. These are segregated early on in development in the somatic and germline tissues. And in the somatic tissue, because the promoter is a germline specific promoter, there's no mobility here because the Cas9 is turned on. But in the germline, it is. And so as this develops into an adult, we see that there could be drive in the germline. There's no drive in the somatic tissue. The target locus is that white eye locus, but we have a dominant copy of the gene, and so we see brown eyes or we see dark eyes. But that comes in from the female. The female now has the drive locus in her eggs. She fills the egg with Cas9 guide RNA complexes so that when the male chromosome comes in, it's immediately susceptible to attack by the guide RNAs. Now, there's no homologue when it first comes in close by enough to facilitate homology-directed repair. So this probably, or could repair itself by non-homologous injoining, just stick itself back together. As a consequence of that, it can make a mutation. And when these then propagate into the somatic tissues and the germline tissues, we now have diploidy restored here. We have the mutation in the female chromosome caused by the drive system, but we also have a mutation in the male chromosome caused by non-homologous injoining. We're missing all wild type copies of the eye color gene, and when we look, we see white eyes, and that's exactly what we see. Now in the germline, depending upon how efficient this is, we still may get drive, because not every one of these undergoes cleavage and non-homologous injoining. So we still may get drive, or it may actually be a drive-resistant allele, and we won't see drive. And when we look in those lines, we see that the drive frequencies get lower, which we did when we saw the data we just showed you. All right, well, what else is going on? Well, we noticed early on when we were collecting insects to do these trials that the white-eyed females that we use to establish the subsequent generations gave very few progeny. And this is a white-eyed female. She's actually a heteroallelic white-eye. So she's got a copy of the drive locus, which is GS red plus, and she's got a drive non-homologous injoining allele because she's got white eyes. And she only gave 200, or this group of females only gave 295 progeny. When we looked at a similar cross, but in this case, there's a wild type allele for the white gene here. So they have dark eyes. And so they're still dry because they're DS red plus, but dark eyes. They had many more progeny. So we asked ourselves, well, what the heck is going on here? And it suggested to us that there's a genetic load associated with making a homozygote in that target gene, the white eye gene. And sure enough, that turned out to be true. And these are a number of experiments where dried mosquitoes were crossed to a very small number of white-eye mosquitoes that we had. And we used wild types as control. we use these heterozygotes as control, and we had the homozygotes that resulted in. The wide-eye phenotype can come about from 3 different ways. It could be a homozygous drive insertion, because that knocks out both copies of the gene. It could be a heteroallelic combination, where you have the drive locus and a non-homologous end-joining locus, or it could be 2 non-homologous end-joining loci. And it turns out that the number of females surviving after a blood meal, so this blood meal is really important, is very low for these wide-eyed animals. And the number of them that survive and lay lots of eggs is actually very low as well. So they don't lay many eggs, and the number of eggs they laid is much less than wild type here. And the surviving larvae then are much fewer than what we see from these other crosses. So there's clearly a load associated with targeting this gene. So what's happening? And this is my getting back at immunologists, but it turns out to be a pretty straightforward slide. So this is work done by Pedro Oliveira. It's just spectacular work looking at the consequences of blood being. And it turns out blood's a highly toxic meal to be taken in by these insects. He's done work both with mosquitoes and rodnias. So when a female feeds on a blood meal, she takes in this blood bolus. Well, the first things that happen is the blood's digested, and during the digestion of the blood, you get free amino acids and also the heme that comes out. Well, this heme can catalyze or be a part of a catalytic response to produce reactive oxygen species, which are dangerous to the insect. And also there is this particular target is a gene in the tryptophan biosynthetic pathway, and the precursor tryptophan can cause what's called hyperamino acid toxicity. And basically then this leads to these reactive oxygen species. Here's the pathway here. We see tryptophan going down, and here's our block here. All right. So the product of the tryptophan biosynthetic pathway that's important here is xanthurenic acid. And what that does is able to interact with reactive oxygen species and to a certain extent detoxify this blood meal. So that suggests that it's a lack of this XA that's responsible for this female phenotype. And sure enough, work done in our group by Vanessa Bettina Rojas shows that if you supplement mosquitoes that are wide-eyed mosquitoes down here with xanthurenic acid, you can rescue this phenotype. So ones in the absence of xanthurenic acid die, so we have % survival after a blood meal, and you can see they die here. But if you provide the XA, they don't die. So in principle, well, I'll get to that in a second. So what happens in laboratory cage trials then? So what we have here are a number of experiments where we've taken males that are the transgenic males in competition with wild type males. Here there's 1 to 1, 1 transgenic to 3 wild type males, 1 to 9, 3 replicates. What we do is we put them in a cage and let them have access to wild type females. We give them a blood meal, we collect eggs, and we grow them up to larvae, and we partition some of those larvae to populate the next generation cages. Some we directly screen as we grow them up for development, so 500 we'll take out. And the rest, in conjunction with these, are used to evaluate the population size. And we look for drive efficiency as the frequency of DS reds, and then the non-homologous end-joining resistant alleles are coming, or the wide-eyed ones that come up. Going too fast. All right, so what happens in these cages? So this is a non-overlapping generation scenario, 1 to 1, 1 to 3, 1 to 9. You can see this is a percentage of DS red, and these are the generations, and this is the number of mosquitoes here. And you can see after release, the drive is very efficient. Okay, and it goes up, in at least 2 of these cages, it goes up to what we call full introduction. Every mosquito carrying at least one of the drive genes, and eventually goes to homozygosity. All right, and we do 3 replicates, and this one didn't quite act the way these 2 did, but it worked pretty well. And when we look at a 1 to 3 ratio, which is even less, we also got 2 cages that went to full introduction and then subsequently to homozygosity. When we use 1 to 9, they went up quite well. but then after reaching a maximum above 80%, they started to decline. Now, what's interesting about this is that once they reach this full introduction and go to homozygosity, they crash, the populations crash. And we anticipate that this would happen because if you remember, if you become homozygous for that wide-eyed target gene, there's a load on the female, and that should cause the cages to crash. Indeed, it does. These here, what happens is that we get a full introduction or get close to full introduction, but then we start to get an accumulation of non-homologous injoining alleles, one that are resistant to drive, but are also functional, all right, because they provide that function, and the population stays stable as a consequence of that. Okay, so once they reach this peak, there's a strong selection for resistance alleles that provide function for, and that KH pathway, and the populations will persist. Well, it's possible to rescue that load. So what happens if we do that? And this is work done by a number of people in the lab, but worked up by Adriana Adolfi. And basically what you can do is introduce plasmids that carry 2 guide RNAs that cleave in different parts of the target, in this case, the integrated drive construct, and you supply the plasmid that has a recoded wide-eye gene on it. So it's called KH recoded. And this one is marked with GFP, the original is marked with DS red. And when things work out, which they did, you can get GFP marked mosquitoes with wild-type eyes because they have the fully functional target gene here. And we can show that it rescues that female phenotype because here are wild types, here are homozygous recoded KH genes, and here are heterozygous over a defective target gene. And then here are the 2 defectives here. You can see that the eggs per female in these gene-drive mosquitoes in the homozygous and also the heteroallelic pair here are comparable to wild type. So it rescues that phenotype. And the larvae produced are also the same. So it's possible that by putting in that recoded target gene, that you can mitigate the load and restore wild type function. And when we do cage trials, it's the same sort of thing that we saw before: 1 to 1, 1 to 3, 1 to 9. It's green now because these are green. We can get not only full introduction, everybody carrying a copy of the gene, but homozygosity, and these persist. And they actually work out very well at very low ratios here at 1 to 9. So, so the drive system can work very well under those circumstances, providing a recoded target gene. Well, what happens if we change the target gene? The pathway that we worked on, the tryptophan metabolic pathway, starts with tryptophan, goes through a number of products, and ends up in the eye color pigment here. And we had blocked it in the Anopheline eye at this stage here, preventing the accumulation of xanthurenic acid. Now, changing species here, we decided that when we designed something for Anopheline gambiae, we would actually take the next step in the pathway below xanthurenic acid, so we wouldn't run into that potentially deleterious load associated with the target there. And so with the system, once again, now that looks very much like what you saw before, you have a target gene here in the chromosome, and you have a plasmid which has a construct that's supposed to integrate this. It's a little different than the previous one, that it uses a different developmental promoter called nanos instead of ASA to drive the Cas9 gene. But it does have the U6 guide driving the guide RNA, and in this particular case, it's marked with the blue fluorescent protein. So if we generate now a primary line with this, we can see the blue fluorescence in the larval stages here, and then we can see a phenotype associated with integration of this gene. Now, this is the ortholog of the cardinal gene in Drosophila, which has a specific phenotype, and we can actually see that here. These eyes are much lighter than the wild type, because in this pathway here, you're blocking the xanthomycin pigment. And we can score that in the larvae, we can score that in pupae, and we can actually score it in young adults. But it's interesting, after a day, that phenotype starts to look more like wild type, suggesting that there's another pathway to xanthomycins in the mosquito. But this is very convenient to have these phenotypes in subadult stages and adult stages, and places where you might want to monitor a drive and you don't have access to fluorescent microscopy. We're doing good. And what happens in the laboratory cages, very similar to what we saw with the recoded, we see that we get very rapid introduction in homozygosity at all three release ratios. So this is a very efficient drive. They go to 100%. And the populations generally stay stable as we move along. All right, so this is a complicated slide. I'm not going to wade you through all of it, but the idea here is to address the concerns that wild-type mosquito populations may have target sites that have significant changes or polymorphisms in them, that they would not be a target for the Cas9 guide RNA drive system. That is, that the wild-type target genes may be sufficiently variant that they have naturally occurring resistance alleles. So this is work done by our colleagues who are doing the field work from UC Davis, and they looked at a number of colonized strains, the Anopheles gambiae and Anopheles colucci, looking for variants, and they could find them. And so what we have here are the 23 nucleotides of the target sequence. This is the guide RNA target sequence. This is a domain called the PAM domain. It's the highest in terms of sensitivity to mutation. These two of the three nucleotides are. One of them is not. That there's a seed region and a non-seed. And so we could, the point is though, we could find variants. And so this suggested to us that we needed to investigate these variants. And what we did is we generated a number of oligonucleotides that contain those variants in them. This is a wild type sequence here. This is one variant, another variant, et cetera. And these are found through mosquitoes throughout Africa. But we cloned these variants into a plasmid that could also then be cleaved by MCO1. And when you do that, that linearizes the plasmid, and that's what you see across the top here. And then we incubated this in the presence of the Cas9 nuclease and the guide RNAs and asked, do we get cutting in vitro? And the answer was for all these variants that we do. So here's 1, 2, 3, 4, and 5. And here's evidence that they actually cut. So while these variants exist and are potentially resistant, they in fact are not. Certainly in this in vitro assay. Now, extrapolating this to in vivo is difficult, except for I will tell you that this is the wild type here. All right, and you can see that there's still quite a bit of uncut in the wild type. So this must be a biochemical issue here, or biochemical characteristic here, because we know we get 100% drive. So we haven't been able to figure out whether or not this could actually be translated into some kind of drive efficiency in vivo. But you know, likely it cannot. So, but the point is that the naturally occurring variants we did see were cleavable and therefore should be cleavable or targeted by the drive system. It may slow it down a little bit, but it won't stop it. And that is significant because even if it slows it down tenfold, as you can see here, this is slowed down tenfold just by the ratio of animals that are there. You can still get drive within a reasonable amount of time. The other concern that people are worried about are off-target effects. That is, other sites in the genome that look sufficiently like your target site that they might be cleaved by the Cas9 guide RNA complex and then cause a phenotypic or genotypic and phenotypic effect that may be deleterious. Now, this is much more of a concern of people who are working in gene therapy for vertebrate cells. For us, and I don't mean to sound flippant, but if it kills a mosquito, you know, that's what a whole lot of other people are trying to do. So it's not that big of a deal, but we wanted to look at it anyway. And so we looked at in the Anopheles gambiae genome, and we looked for sites that looked a little bit like our target, or the best sites that look like our target site. And these are called off-target variants. And then we also had a control sequence here where we took our normal sequence and then eliminated the PAM sequence here, and this is important for binding and cleavage, and so this should be a negative control. And we did the same kind of experiment. We took all of these and we cloned them into a plasmid, cut it with NCO1 and linearized it, and then put it in a Cas9 guide RNA in vitro system. And you can see that off-target sites 3 and the 2s here did not cut at all. So there's no off-target cutting for those sites. Off-target 1 did cut a little bit, and we looked at this, and it's extremely infrequent in vivo. Off-target 1, off-target 3 are sites actually within coding sequences of genes, and off-target 2 is in an intergenic region. And when we looked in our laboratory strain, it actually had 3 different variants naturally existing in our laboratory strain. So we had off-target 1, 2, and 3 here, but none of those cut as well. So we don't see significant off-target activity as a consequence of this. All right, so I hope I convinced you that these drives look pretty good. So the question is, how good do they actually have to be? And then coupled with the effector molecules, how good do they have to be? So they must meet sufficient, they must meet efficacy and safety criteria. And at the same time, as a product, they have to have components that address regulatory and social criteria. So this led to the development of what are called target product profiles. And as we start doing this, we're no longer in an academic environment. We're talking about developing a specific product. And the target product profile has ideal characteristics, and this is what most of us think about. You know, if it's perfect, it'll do this. But it also has what are called minimally essential, which tends to diminish this concept a little bit. But what this means is that, you know, is it good enough to work, to actually help? So what's minimally essential to get it out there? And this is informed by modeling and also by empirical experiments. And there are a number of modeling papers that have come out recently that help address this. And we have work in our group by John Marshall, who's also working on that. But we developed what's called the target product profile. And the question is, how well does this Gambi product, for example, meet target product parameters? And we don't have time to go through all of these things, but source DNA size, complexity, stability, drive target locus, competitiveness, fecundity, strain maintenance, all of these things here, inheritance, timed introduction, off-target non. This Gambi construct meets every one of these. So it's made this checklist. And so we're very happy with that, the performance of that. So in order to get this out of the lab into the field, we of course need to couple it with effector molecules and finish up the lab work, which is phase 1 work. And phase 2 basically could be either contained or in the field, contained field tries, or ecologically constrained. And we're interested in this pathway here. All right. And should our products meet the phase 2 challenges, then we hope to go to a stage release. At that point, it's an implementation phase. And all of this is covered by, or accompanied by regulatory oversight, community engagement, and communications. So this is the pathway that we're envisioning for moving out. So to do this, we have what's called the University of California Irvine Malaria Initiative. And right now there are 5 components, 4 UC campuses and Johns Hopkins. And the mission is to promote the discovery and development of novel science with the goal of malaria eradication. And we have a field site component that's being run by the Vector Genetics Lab at UC Davis. And they developed a volume which addresses site selection criteria for Africa. And they divide it up into tiers. The first tier to satisfy is that, you know, we're going after Anopheline Gambi, and we would like to see geographic and genetic isolation. If we can find places in Africa where these are met, then we go to tier 2, which is island topography. We need to know about insecticide resistance. Can the strains, wild strains, be colonized? What's the plasmodium prevalence? And if we can meet these criteria, then we go to a tier 3 criteria, which have to do with security, willingness to collaborate, human resource, etc. So you can see these things that they fill out. So once again, it's a phased approach to move us from the laboratory to the field. But in this particular case, we're focusing on the field components. And they performed this exercise and identified 2 potential sites for further development on the west coast of Africa, the island nations of São Tomé and Principe, and on the east coast, the unit of Comoros. And these meet some of the geographic features. This is São Tomé Principe. The closest mainland is Gabon, which is 304 kilometers apart. The two islands are separated by 140 kilometers. They have geographic features to them. And we were just getting ready to go there and collect more mosquitoes when all of us got grounded. So while we have agreements with the government and the universities there, nobody's doing anything at this point because nobody can travel. Similarly, the features of the Comoros are that it's off the coast of Eastern Africa. Tanzania is quite a distance away. Madagascar is quite a distance away. There's distance between the islands, and there's preliminary analyses show genetic structure of the populations of individual islands. So these represent potential isolation spots. And once again, we plan to go there. The group had planned to go there, and we're waiting for the epidemic to abate. All right, so what are we seeing? We think population can have a modification, have a role in malaria eradication agenda. We've seen proofs of principle for laboratory and drive components that have been demonstrated. We think the cage trials are extremely necessary. The small cage trials are necessary for evaluating the product at the preliminary stages here. And the sites for testing, evaluation of these sites is in progress. So this is the last two slides here. This is to say that we don't think that population modification alone is going to be sufficient for malaria eradication. We think that all these components on this wheel here are going to be necessary. Environmental management, drugs, vaccines are on here somewhere. Yeah, monitoring, all this stuff is going to be necessary before we can actually achieve eradication. But we do believe that modification can have a significant role, as we indicated, in maintaining local elimination and then leading to eradication. And I will stop there.
Thank you, Tony. This was really, really good.
Yes, it's true.
I think people who want to make questions, you can turn on your video. It's nicer to interact with someone with a face. And you can, I think, either make the questions directly through your microphone to Tony or type it in the chat window.
Bravo, Tony.
Oh, thank you. Thank you.
I have a question, Tony.
Please.
So, are you considering the inactivation or knockout of parasite host factors as a way of blocking the parasite? And do you think a strategy like that would be easier to integrate with a gene drive because you're not carrying a cargo, you're not carrying, you know, a transgene?
Yeah. So, so the question is, can one identify a host factor in the mosquito where if you knock it out, then it impairs the transmission of the parasite? And the problem is that we don't know what that host gene does for the mosquito in many cases. And so when we started out just with the plan of doing this, we decided we weren't going to do that because we didn't want to have to deal with potential loads imposed on the mosquito. Now, we've just shown that with the recoded stuff that one could potentially restore that function, but I can't see how you can restore that function to the mosquito and then deprive the parasite of that at the same time. Okay, so the answer is yes, and I know people are very much interested in trying to do that. That is, it's what's called atreptic immunity, right? Which is to remove something vital for the parasite. But I'm just a little bit nervous, and I would be getting into some pretty fancy biology with the mosquito in terms of what the mosquito wants to do to use that. So our approach was just to give it an extra gene instead of knock out a gene. Yeah. Though having said that, there's some loci, which there are polymorphic variants, and one variant functions well in the mosquito but doesn't do its job for the parasite, and another variant you know, which does it for both. And so I think FREP1 is an example. I've simplified that tremendously, but I think FREP1 is an example of that. And FREP is actually in a pathway. There are multiple genes where the mosquito might actually be compensated by other members of the family, but knocking out a very specific one that the parasite's using would have that impact. And as you know, you are very much interested in FREP1 as we are as well. So we can test that.
Thank you.
I hope that made sense.
Yeah. Can I ask a question?
Yes.
So, I mean, I hate the word eradication, please. There's a long way to go for that. But what interests me, of course, is the diversity in Africa. I mean, you're looking at Gambi, but there's the sibling species of Gambi. You've got Colitzi there. And
right.
Also Phonestus is a big deal too. I mean, to me, this is the right step to go because this is going to take the place of insecticide control. Right. And I think that this is the way we have to go. And I think it's far more ecologically effective, acceptable. So I really am impressed with what you've done. It's great. But I just wanted to know about the diversity of these vectors. Right.
So this is a great question, and so that last slide was also supposed to convey the idea that we don't think that this is going to be a silver bullet applicable everywhere. So it's going to be for those places where Gambi, at least right now, Gambi is present or closely related species. That does leave you with the issues of Phonestus, for example. But one can imagine that once this technology has been made easily done, and I mean, I'm hesitant to use the word easy because you know, people assume you're taking shortcuts. But what I'm saying is once you've laid out a pathway for how to do this, if you have several major vectors, it wouldn't take that long to make a similar sort of thing for Phonestus. It wouldn't take that long to make it for other non-hybridizing species. Now, having said that, I do expect, for example, that if we made something for Gambi, that it would intergress into Colutzi. We want to test that, okay? Because any species, any of the species complex members that form viable and reproductively viable F1 hybrids, so they continue to reproduce, should actually, in this case, now have an introgression of this gene into them. Yeah. So this should hit not just Gambi, but the other ones where you get viable hybrid offspring. But it won't go into things where there aren't viable hybrid offspring, and you'd have to come up with something new for that. Now, having said that, though, it probably would be a whole lot faster and cheaper to make it for Phonestus than it would be to come up with a new insecticide, for example. I mean, I just think it's going to be much more efficient for that. Yeah. Other questions or comments? Oh, I'll look on the chat line here.
I had a question.
Yeah, Connor, please.
How are you? I was just wondering, with the single chain antibodies, whether you'd considered different heterologous strains of Plasmodium falciparum, and if you're planning any R&D activities along those lines to find conserved targets.
Yeah. So we have a twofold. I don't know what the word is for it. There are a couple of things that make us really enthusiastic about this. One is that we're using reagents from the transmission blocking vaccine realm, and people working in the transmission blocking vaccine realm have the same concern. That is that if you make a vaccine, you want to make sure that naturally occurring variants are not going to, naturally occurring variants in the parasites are not going to be resistant to that vaccine. So there's a lot of work being done on the epitopes for these single chain antibodies that we're using. And it would have been another lecture today to give you, but we are actively looking at that. We have a collaboration with some folks from Australia who are looking at parasite variation, and they're looking at the naturally occurring amino acid variants, and we want to be able to test our mosquitoes on those strains. So that's thing one. Thing two, though, is that we've also got this dual gene approach. So that the idea is that if done properly, if one gene doesn't do a really great job, but gets rid of most of them, your second gene might be able to come in and clean it up. And so one of the things we showed with that first series of drive experiments is that the drive system could easily carry a cargo of at least 2 genes. So we're happy about it. So that's our approach right now. But yeah, we definitely want to know what the target parasite is when you go into an environment, and show that the effector molecules do their job against that combination or that suite of parasites.
Okay, fantastic. Thanks.
Yeah.
Hi, Tony. I have a question.
Please.
Hi, this is Prakash. You were mentioning, so this is not a question, but rather a comment. First of all, like you were mentioning about how you went, as an assistant professor, you gave a talk ahead of Tony Fauci. I remember giving a talk at one of the garden conferences right before you and Bob Sinden. So that's exactly how I felt as a graduate student. So anyway,
all we share is the name.
With respect to the 2 strategies of inserting your transgenes that you talked about, one where xanthurenic acid production is affected and the other one not. Have you compared the infection rates for falciparum in these 2 strains to discern the effect of lack of xanthurenic acid in one of the strains? Does that play any role in the development of the parasites?
Yeah, we haven't done that, and we probably won't do that specifically. Okay, and I'll just say that the data, so what Prakash is referring to is that there are data that suggest that xanthurenic acid is important for parasitic exflagellation following the blood meal. That's an area where I'm a little bit uncomfortable commenting too much about, because the model system of Aedes aegypti, Plasmodium gallinaceum, there is a XA deficient strain of Aedes aegypti, and it supports the development of Plasmodium gallinaceum very well. Okay, so I don't know what's going on there. It may be sufficient XA is actually provided by the host to have an impact there. I just don't know. But having said that, if it's true that that's happening, we get a little bit of a lift, but we lose that by the recoded one because we actually provide it now back. Yeah, but I wouldn't target that one specifically just because the data are to me, a little bit confusing. And it may be parasite specific, maybe host parasite specific, or there's something else going on. But it's not, for me, not a great target. Yeah. Okay.
So hi Tony, this is Kailas.
Yes, hang on. I've got an online one here. Opielua, did I get that right? It says, how do you overcome the possibility of decreased propagation of the gene drive due to the formation of non-homologous injoining resistant alleles? So there were 2 things there that that we show, and that is that one, that we don't see Cas9, well, I didn't actually show those data. We don't see Cas9 guide RNA generation of non-homologous injoining alleles at very high rates. I didn't have an opportunity to show that. But what we did show is that variants that may be potentially resistant in wild populations still cut in vitro. So it suggests that unless it's a strict resistance allele that had that was at high frequency and had a resistant genotype, this is not going to be a problem for us. And one of the things that we're hoping in terms of our target product profile is that a single copy of the resistance gene will be sufficient to confer refractoriness to malaria parasites, so that you could even be a heterozygote for a resistance allele and a drive allele and still be resistant to malaria parasites. And as long as there's nothing driving the formation of homozygote resistance alleles, it shouldn't be a problem. But we have to test that. And so we're going to try.
So hi, Tony, this is Kailash here.
Yes, please.
So if there is a vivax and falciparum mixed transmission, so how the approach will be?
So our phase 1 here is strictly for Anopheline gambi, but we have in the generation of some of these resistance genes the opportunity to try to test them against vivax at the same time. So ideally, it would be great to have something that works against vivax and falciparum, but minimally acceptable for at least making an initial impact would be sufficient to go up against falciparum.
All right, thank you.
That's my perspective on it, yeah. Other questions or comments? All right, so let me encourage you to, oh, Craig's there.
Yeah, I have a quick question.
Sure.
I'm sure you must have done this, but for your target, for your guide RNA,
Yes.
did you establish that there aren't polymorphisms that you can find in indigenous populations?
Yes.
Yeah.
But those were those, and then we took those and put those in the plasmid and showed that at least in vitro they were cleavable by the Cas9 RNA complex. So that was that image, and so how that the predictive value of that for in vivo is that, well, if there is an impact, the best it would do would be to slow it down. Okay, so we would see it extended over longer generations, and you know, so that's the prediction. So that can be modeled. But yeah, we haven't found any of the naturally resistant, the natural variants that are resistant to the Cas9 guide RNA cleavage.
Got it. Thank you.
Do we have any other questions?
I know it's Friday night there, but no one's going anywhere, right?
That was the applause. Well, thank you so much, Tony.
You're welcome.
It's been a wonderful seminar. We had close to 150 participants.
Wow.
It's been really nice. And tomorrow is World Malaria Day too.
Yes,
so
exactly.
this was a very good coincidence.
Yeah, yeah.
Thank you.
Thank you, Tony.
Yeah, thank you. I appreciate it. And there's a few more. Thank you.
Thanks, Tony, very much. It was very good.
Yeah. We'll do
Good job, Tony.
all right. You guys take care.
Yeah, and this presentation has been recorded and it will be uploaded through our JHMRI website. So if you need information on where to find it, email Genevieve or me. Thank you. Thank you again. Thank you, everybody. Thank you.
Thank you so much.
Pedro.
Nice to see so many familiar faces.
It's crazy, isn't it? Wow. This is nicely done. Thank you, Genevieve. Thank you, George and Fotini. It was good.
Thank you. Thank you, Tony. Thank you, everybody.
That's the longest I've talked for a stretch in a while. So
it went really well.
Tripping over my tongue there.
Oh, yeah, you went fast.
Genevieve for getting me on here.
Yeah. Can keep me on the list because
oh yeah.
You know, I know you're going to try more of these, and it'd be great to be one of the faces in the crowd at that point. I would recommend getting Greg into too to talk about the field stuff because there's just a lot going on with that. And you know, I didn't do the nuts and bolts because of a lot of the stuff because it just would have taken hours. So
yeah, but make sure Greg gets on.
Yeah. All right, you guys take care.
All right, take care. Take care. Bye bye.
Thank you.
Bye bye.